So here we are in our third lecture, and in this chapter, chapter 3, Cells, the Living Unit, we look inside of our structural units of all living things, and cells are that smallest unit of life. We could have single-celled organisms and then us, multi-celled organisms. So we're going to focus here on structure and function and specialized cells, and do a little bit with just genetic information, DNA and RNA for protein synthesis. So we begin with the idea here that the cell is the smallest living unit. Regardless of form, regardless of behavior, we have this microscopic package containing everything needed for survival. And we rely on homeostasis. So the loss of homeostasis is going to underlie all the things that go wrong in our body. And we have this late 1800s research that provided us with what's known as the cell theory, right? So our cell is the smallest unit of life. All organisms are made of one or more cells, and that cells arise only from other cells, so via mitosis, which we'll talk about later. We have three major parts to the cell. We're going to talk about each of them. We have a cell membrane, which we'll talk about first. We have our nucleus, which we'll talk about second. And then we'll talk about the cytoplasm and all of its organelles before we switch gears into transport and how things are moving around, and mitosis, DNA at the end. So we start with a discussion about our cell or our plasma membrane, and the job here is to separate two fluid compartments, intracellular fluid within cells and our extracellular fluid outside of cells. So nearly all of our cellular organelles are enclosed in a membrane, and so we start here. We have a selectively permeable membrane, meaning that not everything gets to go through the membrane. Okay? It's made up of phospholipids and proteins and cholesterol. So we have this double layer, right? So this fluid mosaic model with a double or phospholipid bilayer. And what's interesting about this is you get polar hydrophilic heads, which are attracted to water, and then your non-polar hydrophobic tails that line the center. So that gives this fluid model of a thin double layer. And then we get some structural support from cholesterol, which is going to decrease membrane flexibility, and it's about 20% of the membrane. And it's got a kind of a similar bilayer. And so we'll talk a little bit more about this idea of our non-polar or hydrophobic tails lining the center on the next slide. In your book, I think somewhere around page 64, you'll find this diagram of the plasma membrane, and it talks about the three components that are contained in the membrane. So you've got lipids, proteins, and carbohydrates. The point of the hydrophobic core is to restrict passage of water-soluble molecules in and out of the cell, allowing some polar molecules to pass. And so we've got membrane proteins for signaling. You got cell surface markers that are like a name tag, and they're there for structural support, transport, control of chemical reactions. They're a receptor site for hormones, antibody production. We've got a lot going on. We've got our membrane carbohydrates, so adhesion of cells, only found on the outer surface, whereas you can get a lot of different things going on with the proteins. And then with the lipids, we already kind of talked about cholesterol and your phospholipids when we're talking about the structure itself. And again, check out the information There's a lot of really good information in your book with focus figure 3.1 from that textbook or your e-text, and it talks about the fluid mosaic model and adding key players and how things are moving in and out. And we'll talk about it a little bit more with transport. Most of our cells have at least one nucleus. So things like our red blood cells are anucleate, meaning they have no nucleus, and they live for like 3 to 4 months, and then they deteriorate. Some of our cells are multinucleate, like your skeletal muscle cells or bone-destructing cells, and these have many nuclei, and then they're typically, larger than usual cytoplasmic mass until they have more. But the nucleus in general is a large sphere in the center of the cell that contains the DNA. It sort of acts like what we would kind of consider the brain, meaning that it's going to control cellular function. And it has 3 structures. We've got a nuclear envelope or nuclear membrane, we use those terms. And this is a double membrane barrier separated by a fluid-filled space, kind of similar to what you see with the cell membrane. The outer is going to have rough endoplasmic reticulum, and the inner is going to have some rod-shaped proteins. That's kind of keeping the structure of the nucleus. We have nucleoli, which are spherical bodies where our ribosomal subunits are assembled. Not membrane bound, and we typically have 2 nucleoli per nucleus. Interesting. And then your chromatin, which are our bumpy threads, they are comprised of 30% DNA, 60% histone protein, and 10% RNA. And this is the structure that is our uncoiled chromosome. So before the cell will undergo mitosis, these would coil up into our typical, what you see as a chromosomal structure. And into the cytoplasm we go. Cytoplasm is a semi-fluid cellular material between the cell membrane and the nucleus, and I just put a very basic picture there. And the cytoplasm is going to be home to all of our organelles, and our organelles are specialized structures that are going to perform specific functions within the cell, and we're going to go through a lot of these. Right? So this is just a semi-fluid filler, so to speak, but it's going to house a lot of what does the work for the cell. So we'll start by talking about the mitochondria. The mitochondria is the powerhouse of the cell, producing our ATP. If you have more mitochondria, you're typically going to see more activity. So think about a type of cell in the body where you would guess you would have more mitochondria because you need higher activity, and I'll let you just think on that. We have ribosomes, which are granules that manufacture or synthesize our proteins. They come in 2 forms. They could be free or they could be membrane bound. And in this picture you see membrane bound ribosomes on our rough endoplasmic reticulum. Endoplasmic reticulum is our fluid-filled cisterns, essentially, that help build cell membrane, help build proteins, help build any other materials that are exported from the cell. The way to tell the difference between rough ER and smooth ER is that rough has ribosomes. You can have a little fun with that. So the rough have ribosomes, so they are the site for protein synthesis, where your smooth that do not have ribosomes are site for enzyme reactions, sex hormone production, and the synthesis of cholesterol or fat-based substances. So 2 different types of endoplasmic reticulum. I showed you just a picture of the rough with the ribosomes on it so you can kind of get a sense of what that was going to look like. So once we've synthesized some proteins, we also have to kind of modify, sort, and package them for specific distribution. So the Golgi apparatus are stacked and flattened membranous sacs that are going to kind of determine, like a traffic director, what needs to happen with the proteins that have been synthesized and distribute them via exocytosis and they can be set up for storage within the cell or shipment out of the cell. We have peroxisomes, which contain powerful enzymes. They break down and synthesize fatty acids. They're pretty numerous in the liver and the kidney, and obviously active in detoxification, right? Because we're talking about breaking down and synthesizing fatty acids. We have our lysosomes, which are the digestive system of the cell. They're going to bond with food vacuoles, and they carry digestive enzymes with them. A little bit different look at a lysosome versus a peroxisome, and understanding that peroxisomes are dealing with fatty acids, where our lysosomes just general digestive enzymes and some other focus, so that they are a little different. And then in order to give all of this some shape and support, we have what are called cytoskeleton rods for support. So these rods will form the supporting structure. We have microfilaments, such as actin, and we have microtubules, which you find near the nucleus, and those are different. We talk about actin and myosin in the muscular system, so we'll come back to what's going on as far as microfilaments later in the muscular system. And to round out the organelles found in the cytoplasm, or specialized structures in general of the cell, we have microvilli, cilia, and flagella, and they are different, so I've tried to include some pictures so that you can see what's going on. Microvilli are folds that help to increase the surface area, so you'll see this in the small intestine in 202 for sure, with a brush border. And then we have two structures that are used for different types of movement. Cilia are hair-like, and they're for essentially passing things over the surface of the cell, whereas you have a flagella, which is a whip-like structure to propel cells. So you may see a whip-like structure, like a flagella, on a sperm cell, right? And cilia you'll just see in general, potentially anywhere, things you can move over the surface of the cell. So our selectively permeable cell membrane is going to have things that it allows to just move through, and some things that require assistance to pass through, right? And assistance is categorized as non-carrier mediated or carrier mediated. What we're talking about there is dealing with whether or not it can pass through, or if it requires a membrane protein specific for transporting some polar molecules or classes of molecules like sugars and amino acids that are too large for just a membrane channel. So the difference between passive transport, where we do not require energy, and active transport, where it requires energy to move substances. So we're going to start with passive transport, and then we'll talk about active transport after. So passive transport is all about moving down the concentration gradient without energy from essentially an area of high concentration to low concentration. And lipid solubility and size are going to kind of determine the ability to cross the membrane. We have 4 types of passive transport that we mention. 3 are most commonly spoken about. First, we have simple diffusion. So this is our unassisted scattering through the membrane. This is non-carrier mediated. Typically, you're talking about things that are small and non-polar and that dissolve readily in lipids. Things like oxygen, carbon dioxide, steroid hormones, and fatty acids. Again, we're talking about moving from an area of high concentration to low, down the concentration gradient. Second, we have facilitated diffusion, and I've included a graphic here to help you understand simple diffusion versus facilitated diffusion. Facilitated diffusion is carrier mediated, so there is a protein channel as a transport vehicle, right? So that's your protein channel, as shown in the picture, is your carrier. So your molecules are going to bind, polar molecules are going to bind to carrier proteins, and then they're ferried or moved through water-filled channel proteins, or ion, could be ion as well. So this one, while going from high to low concentration down the gradient, does require a carrier. Filtration is the movement of solutes from high to low. It is a passive process, but it forces fluid through the membrane, driven by a pressure gradient. So we're still going from high to low, and think of similar to what's going on with capillary walls and blood pressure as a type of prescription. And then we round out our 4 types of passive transport with osmosis. Osmosis is specifically the diffusion of water, and then you'll see how that works with a solvent in the picture, from high to low. This is non-carrier mediated, and we have 2 things to consider here. First, we have osmotic pressure needed to prevent osmosis, and it's the measure of the strength of the draw of water. And we have osmolality, which is the measure of a fluid solute, right? So whereas diffusion is solute molecules move from high to low concentration, it's essentially just the equal scattering. So if you put drops of food coloring into a glass of water, right, it starts out in one spot where your drops hit. Eventually, the whole glass is essentially equal concentration. Osmosis is solvent molecules move from low to high solute concentration, and you end up with the same concentration, right, but not necessarily an equal diffusion, right? So they're not distributed equally. So for example, I put an example in here. If you get increased osmolality, so your blood increases osmolality with dehydration. So let's focus on that one. So we're dehydrated. So we're going to increase osmolality, which is going to increase osmotic pressure, which is therefore going to increase the desire to draw in water. Okay? So you get dehydrated, you have this desire to draw in water. That's very broad, very basic, but that's the general idea. So as you increase osmolality, you have an increase in osmotic pressure, and you have a desire to draw in water. So we have some factors that determine speed of transport. So remembering that rate can be based on surface area and membrane permeability, but all with those considered equal, right, we have 3 factors. So if we have a greater difference in the concentration of diffusing molecules or ions between the 2 areas, we're going to have more collisions, and diffusion will be faster, right? More collisions, faster diffusion. So if there's more present, they will run into each other more and therefore diffuse faster. Molecular size or molecule size, smaller molecules are going to be able to pass faster. So smaller molecules move more rapidly, so the speed of transport is faster. Then, finally, with temperature, at a higher temperature, there's more kinetic energy, so we're going to increase the speed of molecular movement, and therefore we're going to have more rapid diffusion. Hopefully those make sense, right? So everything I posted obviously was how you get a faster speed of transport, but obviously the opposite would be true and slow down the speed of transport. And finally, with transport, we talk about tonicities, or the ability of a solution to change the shape of cells by altering internal water volume. We have 3 different tonicities to consider. The first is our isotonic, and if you look at the basic graphics at the bottom, it'll kind of give you a little bit of understanding of where solutes versus water are. Isotonic has to do with equal movement of water in and out of the cell. Therefore, there's no real osmosis going on per se. The cell will be of normal shape. There's no loss or gain of water. We have the same osmolality as plasma. So you can see in that middle picture, you have equal concentration solutes and water in and out in that picture. With hypertonic, we have a higher concentration of non-penetrating solutes. You can see more solutes on the outside than on the inside. So cells in a hypertonic solution tend to lose water, and the cell is going to shrivel up or crenate, for example, with saline. So we have an increase in osmolality from the plasma here. Again, more concentration of those non-penetrating solutes. Water is going to try to equal that out, thinking back to the picture I showed you on the previous slide. More solutes, right? Less water inside because it's trying to create that equilibrium of the amount of water and the solutes. So the cell shrivels up, crenates. And then in our hypotonic scenario, you can see here, we have a more dilute situation. So we have fewer solutes outside, more solutes inside. So we're going to be gaining water to balance this out. So we're going to get a hemolysis, or the swell of the cell as the water rushes in. This could cause cells to burst. So we have decreased osmolality plasma. And you see this scenario in distilled water when you do a little quick lab with salt water versus distilled water and cells. As we switch to active transport, we remember here that now we're requiring the use of energy or ATP, and these are all carrier mediated. They're moving against the gradient. So these are large materials that can't dissolve in the fatty layer of our membrane. Kind of two categories, active transport and vesicular transport, and we'll talk about vesicular on the next slide. But first up, remember, we're kind of going uphill. So if I were to kind of create a fun comparison of passive transport and active transport, I would use an escalator. Escalators are a moving stairwell. So in passive transport, you would step onto the escalator and ride it in whatever direction it's going. Okay? So we'll say you step on the escalator and you ride, it's going up, and you just follow where it carries you. In active transport, you would be going in the opposite direction. So it'd be like trying to run up the down escalator or run down the up escalator, either way, where you are physically having to do work to move in opposition to the normal flow. So here, this is all about moving kind of against the gradient. So we start with our primary active transport. So we get energy direct from the hydrolysis of pumps, for example. So our sodium-potassium pump, right? So the sodium-potassium pump is going to move sodium using energy of ATP hydrolysis to establish a concentration gradient. That's our primary. In secondary, we get sodium moving with the established gradient to drive the transport of glucose against the gradient. So you get energy from pumps in primary, energy in the concentration gradient in the secondary. So transport proteins are energized by transport of the phosphate from ATP in your primary, and in secondary, we're driven by energy stored in the concentration gradients of the ions themselves. We get the sodium pump and then the diffusion of sodium back into the cell after primary active transport, and it's providing power to move a different ion instead. So here we talked about sodium and then glucose. That's this example here. That's a pretty good example. I think you've got other examples in your book, or a better picture where you can see this exact picture more clearly. And on our final slide on transport and cell movement, we finish up talking about vesicular active transport. So here we have fluids with large particles and macromolecules transported across the cell membrane in bubble-like stacks, kind of aka a vesicle. And you have two you have one that's going to move substances in and one that's going to move substances out. So the way to remember this is endocytosis is going to enter, and exocytosis is about exiting. So N-E-N, enter, E-X, exit. That was always kind of a quick trick to help me remember which way I was moving. So endocytosis is the movement of substances from out of the cell into the cell, and we move bulk solids, mostly macromolecules. We have 3 different ways that our substances can move in. Phagocytosis, cell eating, so they're going to engulf materials that bind to the cell surface. They typically fuse with a lysosome and move it in. In penocytosis, this is fluid phase endocytosis, so things with like your extracellular fluid with dissolved molecules. And then we have our receptor mediated, which is most of your macromolecules move this way, things like enzymes, insulin, LDL, iron. But you also get things like the flu virus and diphtheria and cholera that'll hijack this process and take over. So you have to kind of watch that one. And then exocytosis is our exiting. So think of this like waste ejection, so movement of substances from within the cell out. So hormone secretion, neurotransmitter release, and waste ejection. And so it's not just waste, but the big time focus here is on waste ejection. And now we get to head into the last stretch of this 3rd lecture in our semester, and we get to talk about the cell cycle. And I actually truly do enjoy this, and there's so much going on here, more than I'm going to even touch on, and looking at how cancer is impacted and some of those things, or how cancer impacts the cycle, and so many other things. But we talk about the cell cycle as the series of changes a cell goes through from formation to reproduction. It's the cycle for growth, repair, and replacement of the whole cell. And it is controlled by internal and external factors. So we understand that we have things like the ratio of surface area to volume, we have chemical signals like hormones, and we have available space that plays a role in this. The cell cycle is made up of 6 total steps. We're going to talk about some of these. We have our growth phase, which is interphase. This is cell formation to the start of cell division. It's our growth phase. It's made up of 3 subphases, which you can see in this graphic. And really, the cell spends most of its time in interphase. Then we have the division of the nucleus, which is actual mitosis itself. Division of the nucleus is prophase, metaphase, anaphase, and telophase. I'll tell you a little bit more about that on the next slide. And then when we actually finish dividing the cytoplasm and dividing the cell, our final phase is cytokinesis. So the whole thing is the cell cycle. Cell division is the division of the nucleus and the division of the cytoplasm. And mitosis is just the division of the nucleus, which is the 4 phases in the middle. So you got to think about how they overlap, right? So cell cycle, 6 total steps. Cell division includes everything but our growth, our resting, our interphase. Cell division, remember, division of the whole cell. Some of these are active like your whole life, and some have a limited life. So your whole life, things like skin and intestinal lining. Some with limited life, liver is flow, nervous, skeletal, cardiac. Once mature, they don't divide. And then we get into the division of the cell itself. And then finally with mitosis, you're just talking about having that DNA make its copy. Okay, so remember here I'm talking about division of the nucleus and then division of the cytoplasm and the cell itself. So this is cell division, which the first portions of that are mitosis. So we were hanging out in interphase. We went through the 3 subphases, right? So we have a growth phase, a DNA replication phase at the S phase. So at the S phase, Make sure you know that. I think that's a quiz question. So S phase is DNA replication, and there's a restriction checkpoint. After the S phase, we go cyclin, which turn on and off this process. And then in G2, we kind of have one more checkpoint before we head into mitosis, kind of checking that DNA integrity. So I think of this middle part of this graphic as kind of this true mitosis. In prophase, we're preparing. In metaphase, we line up in the middle. In anaphase, the chromosomes pull apart. So prophase, prepare. Metaphase, middle. Anaphase, apart. Telophase, 2 nuclei. Cytokinesis, 2 cells. So let me kind of walk through this one more time because there's a number of questions that show up throughout, I think, both the quiz and this lecture test number 1. Interphase is made up of 3 subphases. Make sure you get to know those. G1, growth. S, DNA replication. And G2 is enzymes and protein synthesis. We have checkpoints between each of those. There's a restriction checkpoint after G1. There's a cyclin protein turn on off checkpoint after S. And after G2, we get DNA integrity checks. Then we head into prophase. You can see prophase in this image here. We are preparing. The nuclear envelope is going to break down. Spindles are going to connect to the chromosome, the coiled chromatin, and they coil up at the centromere, and there's a lot more you learn in some more biology. And the chromosomes are drawn toward the center. So you can see in late prophase there, we have our spindles, they're attached, and they're getting organized. And then as we head into metaphase, the centromeres align along the equator. So they're all nice, pretty lined up right in the middle, so that in anaphase, they pull apart. The chromosomes are pulled apart at the centromere and pulled toward the poles of the cell. You can start to see the cell change shape a little bit. As we move into telophase, we have our 2 nuclei, right? We've pulled that toward the pole. You can see 2 distinct nuclei. Chromosomes are going to begin to uncoil. The nuclear envelope is going to surround each one. The spindles will start to break down, right? We have 2 nuclei, but we're still here in one cell. In order to finish the process, we have to go into cytokinesis. In cytokinesis, we have the division of the cytoplasm via cleavage. So the cytoplasm pinches inward, pulling those 2 full cells from one into 2 daughter cells. Right? So it isn't until cytokinesis where we go from one actual cell to having 2. All of these stages are the cell cycle as a whole. Cell division is prophase through cytokinesis. Mitosis is prophase through telophase. Because remember, mitosis is division of the nucleus. And we finish cell division by going through cytokinesis. So making sure that you understand prophase is preparation. In metaphase, they line up in the middle. Anaphase, they pull apart, A for apart. Telophase, 2 nuclei. Cytokinesis, 2 cells. So I know in your notes it gives you the comparison of DNA and RNA first. That's a pretty inclusive list. I'm going to talk about it again on the next slide. So I went a little bit out of order. And that's because I want to kind of bring your attention to a whole series of events known as central dogma. And DNA replication is one of 3 processes that can go on when we talk about DNA and RNA. So first, I want to talk about just DNA replication. Then we can talk about the difference between DNA and RNA. And then we'll talk about protein synthesis, which comes from the other 2 processes. So central dogma is 3 processes. DNA replicating, so DNA making more DNA in the nucleus. Transcription, which is when the DNA is used to make RNA in the nucleus. And then translation, where we bring up our chain of amino acids, and it's done in the cytoplasm at the ribosome. so using ribosomal RNA in the cytoplasm itself. So first up, the first process here is DNA replication. The hydrogen bonds are going to break between the bases, and helicase enzymes are going to uncoil or unwind the DNA. The two strands are going to separate, and where they unzip is known as the replication fork. So right now we have two strands present, right? Two original DNA. And then the old strand is being used as a template, and another enzyme, DNA polymerase, is going to match our free nucleotides to form the new strand. This is a semi-conservative process because your two new strands of DNA are going to be made from the original. So you have half of it as an original and half of it as new, so it's semi-conservative. You get half old, half new to equal the new molecule. So it's semi-conservative. We're not just starting from scratch, we're using a template to build it. Okay. And then a ligase enzyme are going to splice it back together into its double helix. So we're going to uncoil, separate, assemble, and restore. And that happens using the original DNA strand as a template for the two new strands. So far we've just talked about DNA and DNA as a double-stranded structure, and this table gives you a little bit more, I think, than you see in your notes, but this is kind of giving you the basis of understanding for how it's designed and the base pairing. And DNA is going to be responsible for the first part of protein synthesis, or the second step in central dogma known as transcription. And we have to use the DNA, and then it has a slight variation in the RNA. So you can see that the 4 nitrogen bases used in DNA, A, G, C, and T, right, are changed slightly in RNA to be A, G, C, U. The DNA is always going to stay safe in the nucleus, whereas your RNA can be created in the nucleus, but then can travel into the cytoplasm, and your DNA is not going to go into the cytoplasm. DNA is involved in transcription. RNA is directly involved in both transcription and translation. And while we only have nuclear DNA and mitochondrial DNA, we have multiple types of RNA, and mostly we talk about mRNA, tRNA, and rRNA, but there are some others, and the more we learn, the more we get the fine details. And you can see the base pairing rule kind of listed there. So protein synthesis has 2 phases to it, the second and third phase of central dogma. So we start with transcription, where your DNA contains the blueprint for RNA. So production of transcription occurs in the mRNA in the nucleus and requires an RNA polymerase, RNA polymerase. And we're going from DNA to RNA so that the RNA can leave the nucleus. And it's only a single strand or a half of what the DNA was. So we're transcribing the message, making a copy. So we make mRNA from DNA, it detaches, it leaves the nucleus via a nuclear pore headed for the ribosome. In translation, we're making sense of the message, right? So transcription, we're copying the message. Translation, we're making sense of the message. It's a little bit like playing telephone, except we have a lot of checkpoints and the message hopefully doesn't get jumbled by the time it reaches its destination. So translation is done by tRNA with the support of rRNA at the ribosome, and it's going to create a polypeptide chain that becomes the protein, and all sorts of things are going on. So we're connecting amino acids brought by tRNA to decode the message to build the protein, right? So we're going to scan our mRNA for a start, so we have initiation. We're going to move in one direction, adding amino acids for elongation, and we hit a stop codon at the end to release the protein known as termination. And I'm going to show you a little bit more about that on the next slide. So here's those steps again, right? We have the unwinding of the DNA, the matching of mRNA codons, transcription occurring in the nucleus. The transcripted RNA leaves that segment of the DNA and heads for the ribosome. The tRNA is going to contain the anticodons, which give you the specific amino acid. So info is passed from codon to anticodon in translation. So transcription is about making the copy. You can see that, making the copy of the DNA in the nucleus. And translation is about making sense of the message at the ribosome in the cytoplasm so that we can actually get our amino acids and release our protein when we're done. And this is the end of this segment here. Just close with a couple thoughts on the final slide. So in closing, just to recap, we went over three main parts of a generalized cell: the nucleus, the cytoplasm, and the plasma membrane. The cytoplasm housing all of our organelles, specialized structures for activity, the nucleus being the brain, and the plasma membrane controlling transport. We discussed the difference between active and passive transport and a number of different methods for transport. And we finished with from DNA to protein. So we get the transfer of information that goes from DNA base sequence to complementary base sequence of mRNA, and then to the tRNA base sequence. So from the triplets of DNA to the codons of mRNA to the anticodons of tRNA, which is mostly matching except for uracil instead of thymine, and we build our amino acid sequence, our polypeptide chain. As far as some other information, there's all sorts of developmental aspects, cell differentiation to consider, and cell aging. Some interesting things that go on with cells, like progeria, a rare disease that mimics aging caused by a defective protein in the nuclear lamina, and it gets kind of like displaying slow growth, thinning hair, brittle bones, arthritis, cardiovascular disease, starting at age 2, and most don't live past age 20. So there's lots of things that can go on in your cells. And, as far as the quiz, there's lots and lots of questions on the quiz with structures, the difference between passive and active transport, understanding what's going on with your tonicities, and then the steps for central dogma, the comparison, DNA to RNA, understanding the difference between mitosis and mitosis being part of the cell cycle. And I think I carefully mentioned what the S phase is for, so DNA replication. And as always, if you have any questions whatsoever, we have that open chat online. So hopefully this closes out lecture 3.